So we did mitosis, correct? We did the cell cycle. So let's go over, review something about the cell cycle. So in eukaryotes, we're talking about here eukaryotes. Prokaryotes do not have a nucleus. So that's not an issue. But in eukaryotes, the cell cycle is pretty standard. You have, well, it's a cycle, so it's a circle. You have G0, which is, it's out of the whole cycle. It's doing its business without having to do all the other phases. So that's why we call it G0. They have G1, where it's growing, getting ready to do what it needs to do to divide. So in G1, it is growing, it's duplicating its organelles, it's doing everything except for duplicating the DNA. There, it's going to have, duplicating the DNA is going to happen here. Does anybody remember what it's called, this phase called? What this phase is called? Not G2. This phase where the DNA is replicated, where it's copied. No, there's not transcription or translation. RNA is made in transcription. Proteins are made in translation. The phase of the cell cycle where DNA is copied, we're synthesizing DNA. It's okay, it's been a couple months, but really I want to emphasize this again. I keep saying it to the point where it gets annoying, and you have to review your notes constantly. So this is called the S phase. In the S phase, you are copying the DNA. Okay? Then I'm going to move this so I can give myself a little more room. Then after you're done copying it, you have to continue, you have to check and make sure that everything's working well, and that's where you're doing a G2. You're double checking that you have as much material as you need to make copies. And the examples I used earlier on in the year is, look guys, if you're moving out of your house and you're moving into your own place and your mom and dad are moving into another place, then you need to make sure you get your own stove, you need to make your own bed, you have to have your own dresser, correct? You have to double everything you have so that you can move into your own space. Does that make sense? You also have to package everything I said. You're not going to just start moving one sock at a time to the new place. You have to package it and put it in boxes and get it ready to go. Correct? That's what the cell is doing. The cell in G1 is getting new resources, gathering its resources, doubling up everything, the membrane, the Golgi body, the mitochondria, the endoplasmic reticulum, the ribosomes, everything needs to go. In S phase, it's doubling the DNA, because if you're going to go from one cell to two cells, you need two copies of everything. Does that make sense? And then in G2, you're making sure you got everything ready to go. Everything's packaged. The only thing left to do now is to divide the nucleus. You have to divide the nucleus before you divide the cell in a eukaryote. So in a eukaryote, this is called the M phase. And that's where previously we looked at mitosis, which is what this is. This is mitosis. So in mitosis, you make an identical copy. So everything is copied. These two cells are the same. That is how, this is the process, mitosis, dividing in the nucleus. Remember, mitosis means dividing in the nucleus. Cytokinesis, I should go ahead and put that in here. Cytokinesis is the dividing of the cell. So the cell actually divides, and then you start the cell cycle again, unless you go into G0. Does that make sense to you, guys? We've gone over this, so hopefully it does. So mitosis, so you started off as a single cell after mom and dad conceived you, right? You have to have 46 chromosomes, and remember we have to have exactly the same genes and exactly the same place and exactly the same number, 2 copies of every gene, except for the cells, the chromosomes on the X chromosome, which is a little weird because males don't have an X. And males don't have 2 X's, they only have 1. So we only have 1 copy of everything on the X chromosome. And females don't have a Y. So they don't have any of the genes on the Y chromosome, correct? So those are the 2 exceptions. But every other chromosome, the rest of the somebody close that door. 22 chromosomes, you have 2 copies of every gene. And I like to use humans as examples here because that's who we are. But this happens with all eukaryotes. Their numbers change.
It happens with plants.
It happens with plants. It happens with everything. Same process happens in every eukaryote. All right. Or similar. If there's a difference, there are some differences, but mainly the same process. Yeah.
So how does it work with animals that don't reproduce to create offspring? Like, I forgot what it's called, but
Asexual division. They just do this. They're all clones. All their DNA is the same. Does that make sense? They're not. They just keep growing. They get bigger, more and more complicated. As they get bigger, different tissues, different organs, they differentiate. But the DNA is the same. Why? Because you're using mitosis to grow. That's why every cell in your body has the same DNA. Because you're copying the DNA and you're not changing it. Could you give?
Yeah, the computer stopped working for some reason.
Is the other one working?
Yeah, but someone was using it. You have 40 more copies left.
Oh, okay. Could someone take those and pass them out? All right. So that's one of the reasons that your DNA is exactly the same in every cell of your body, and that's a good thing. You don't want different DNA in different parts of your body. You don't want your organs to start fighting each other because they don't recognize each other. That's just not a good idea. So, and let's move on. So, let's go ahead and see meiosis. So, what is meiosis and why do animals do it? Not every animal does it. Like you said, all animals do this. Some plants do asexual division. But they still find ways of solving some of the problems that come with that. Even bacteria can have ways around the problem. And I'll describe to you the problem in just a minute. So let's start with the entry ticket question you had today. So we just talked a lot about mom and dad. So and we said, what is the result that we want? Mom and dad got together, and they wanted in the end to have a baby. They want one, I'm just going to draw a single cell because that's what you make is a single cell. And they want inside that nucleus, they want the nice, healthy baby boy or girl. They want this baby to have 46 chromosomes. They don't want it to have any syndromes. They don't want to have any extra chromosomes, any missing chromosomes. They want 46 chromosomes because they want the child to be healthy. Another exception I'll mention is that some organisms don't do it this simply. Corn, for instance, can double their chromosomes and half them and double them. It's just a craziness that happens. But in humans, we want to have 46 at the end, right? So how many chromosomes did mom have? 46. And how many chromosomes did dad have? 46. What would happen if mom's cell, the egg, and dad's sperm, dad's cell, the sperm, came together, combined? What would you have? Would you have 46 chromosomes? I don't understand your math. I have 46. Dad has 46. Mom has 46. You take those two cells, because that's what happens. You take the sperm, enters, puts the nucleus in, fuses with the egg. You've seen the videos, right? You've seen it in middle school. They showed you egg and sperm coming together. If not, I'll show it to you later. It's not really. You can, why don't you use some of that time instead of TikTok, watch a video about sperm and egg coming together. So conception, sperm and egg come together. I'll show you later.
I don't want to.
It's, could you just chill? It's just two cells coming together. So sperm and an egg come together, the nuclei fuse, 46 in one and 46 in the other. They come together. How many chromosomes do they have? I say what? 92, not 46, 92. Well, that's a problem, isn't it?
It's too much.
It's too much. And then those two, if for some weird reason it's able to survive, it can't survive. It's too many chromosomes, too much going on. It's very confusing. Various reasons it won't survive. But let's assume it did by some miracle survive, and that happened everywhere. Then 92 and 92 come together. They meet each other. They fall in love because they both have 92. I don't know why, but they do. And they confuse their sperm and egg. What happens? What's 92 plus 92? 184. That's too many. It gets out of control quickly, doesn't it? So we have to have a way to do what? What do we need to do? Divide what? Divide the chromosomes in half. That's right. Boom. Meiosis is born. That's what meiosis does. Yeah. So let's go over meiosis. You got your activities? You have drawings in front of you? All right, let's go ahead and look at on the board. You have 5 minutes to answer it. It's up in green at the front, please. It's in green at the front on the board. You have 5 minutes. Good morning.
Wait, are we turning it in?
Yes. I want your answers turned in, and I want your own words. I don't want to see, if I see the same answers on two papers, no.
Wait, are we turning it in?
In your own words. Paper, question on the board. Go, answer. Quick.
Is this in the paper?
Yes, there is, but you can get it later. Right now, you don't. Every day we have work. I know, it's so hard. Unbelievable. It's thick. It's super thick. It's the worst. Can't believe it. Can't believe it at work. It's unbelievable. It's supposed to be easy. It's AP. AP means easy, right? No work. Not that much. Not as much as regular. This is too much. I have other classes, you know. I have other classes. Why?
I feel like you've heard that before for some reason.
Yes, definitely. It's clean sizes. There's always one kid I can't put their face to the name, and I don't ever remember if that person was in the room or not. My first period is just one. This class, unfortunately, I can't get your names out of my head. But I try so hard to forget, but I can't. But first period is one kid. I just never remember. That's old, but whatever. Your new one's over here. You have that question's doing like 3 minutes now. So make sure you get it written down. Name, date, whatever you can write in 5 minutes. I expect a really good answer from at least one of you. Is Kason here? Is he going to show up like 5 minutes before the end of class? No.
He's not here.
He's not here. I hate having to change that. Graves, Ganotis, Louise, Dent. Trent was absent. He'll be late, but I'm going to mark him down so you can deal with that in the office. Isaac, Esther. Esther here? Yeah, there she is. White. I don't even know why they still have her on here, but Kimberly's here. They took her off technically, but there's just this dash. It's so annoying. All right, 1 more minute. Heck, you guys came in late. It's not my fault. I literally been sitting here just relaxing, spinning around in a chair. I don't know what you're talking about. Tell you what, you'll have until I start the coffee. That's fair. On your phone. You got what? I didn't hear you. That's why I said you got what? If I heard you, I wouldn't have said that. I don't know why people say that. You heard me. Did I? That's why I said that. Literally, I said I don't understand. Don't talk about it. Just write your answer. If you don't know, that's okay. That's why you got a thick packet. Did you hear me throw knife? That wasn't even shade, it was knife. Oh, you want me to shut up? Sorry. She said shut up, people. Yeah, and you're part of the shut up too. If she said shut up to me, then she means you too, or at least I do. Shut up. All right, turn in. Let's go. Since you're talking, why don't you play? Go. Thank you. Hurry up. Just made that. Is that what made that noise in my mind?
Cameron does that.
Your turn. Paper clip them together, please, and thank you. All right. So I think the answer is pretty easy, especially since you have meiosis right in front of you. So how do we do it? So if you combine these two together, you take, let me start from the beginning, because obviously I need to. I'm going to go ahead and use this, because that way I don't have to draw and you don't have to make fun of my drawings. I know it takes away some of your joy, because I know you enjoy.
No, I don't.
Your eyes and your face tell me everything. You love to pick on me.
I love you.
Yeah, thank you for admitting. So we'll talk about twins, but first let's talk about meiosis, okay? So meiosis and mitosis, pretty much the same process, except there's some changes in meiosis that allow us to get half that DNA. Two objectives. What are the objectives that are reached? Overall, big objectives that nature wants to fix, right? So there's two. One, it wants the same chromosome number and placement of genes in any being that's going to make a new baby. That's so there's no mistakes. Somebody's not born without eyes or whatever. If you need all the genes on every chromosome, and you need 2 copies of every gene except the sex chromosomes. Yes, it happens. Most of them, but as I said before, most of the time it's miscarriage. It's at least a miscarriage because the embryo can't survive. It can't do what it needs to do because it doesn't have enough or has too many chromosomes. Does that make sense? There are a number of chromosomal abnormalities that can occur that the individual can survive. Down syndrome as 3 number 21s. There's 3 X's, 3 Y's. There's all kinds of things that can happen that people can survive. But most of these mismatches end up in cell death. So same chromosome number and placement of genes. Are we all okay with that? So that's number 1 on the objective list. Number 2, mix up the DNA. It is not a good thing to have everyone be a twin. To have cloned humans is not a good thing. If you have clones, something that kills you kills all of us. We don't want that. I think I've used COVID as an example. When COVID hit the world, something like 2% of the people died. That's better than 100%, right? Why did only 2%? Why did some people have no symptoms? Other people had bad symptoms. Other people just completely collapsed and died. Why? Because of mixed up DNA. Everybody's genes were mixed up, and most of us could survive it. Some of us didn't. There have been times in history where most people died, but some people survived. That happens as well. But having variety allows the population, the species, to survive. Are we all okay with that? Yeah. All right, so DNA, mixing up the DNA is a really big point in all this. So you're going to see a bunch of different places in meiosis where that happens. Pay extra attention. So we start with a single cell. Now, I'm not going to draw 23 pairs of chromosomes. Is that okay with you? I'm not doing it. So if it's not okay with you, that's okay, but I'm not doing it. And I'm only going to draw the nucleus because I'm not going to do all the endoplasmic reticulum and et cetera. So this is the nucleus. So So when we talk about chromosomes, and so here in the I'm going to draw 2 chromosomes, one from mom and one from dad. Again, there's 23 pairs, so you actually have 46 in here. I'm not going to do that. And let's just go. I'm going to make up the A. So there's a big A here gene, and there's a little A gene here. And then down here, you have a big B, and here you have a big B. So 2 genes, 2 different genes, random genes, that control two different things. Are we all okay with that so far? No confusing points? Well, remember what the cell cycle is. Remember in the cell cycle, in G1, we make copies of everything. In S, what do we do? We make copies of the DNA. Now, in the end, we're going to want to make sure that you move one copy that you have. You're not just separating, that's not randomly taking, like, you don't, imagine pouring out a bag of Skittles on a table and you're dividing it in half. Well, most of us would just count them and just split right down the middle, half on one side, half on the other, and that's it, right? That doesn't work with DNA because you have to make sure you have 2 reds over here and 2 reds over here, 2 blues over here and 2 blues over here. Does that make sense? You have to divide it evenly because of our number one objective, which is to make sure you have the right number of chromosomes in the right place at all times. You don't want to mix things up. You want to have 2 copies of the brain gene and no copies of the heart gene. Well, you're not surviving, right? So you have to make sure you're dividing everything properly. All right. So the way we do it is we stick the 2 copies together while we're copying them. We copy them, which we'll do DNA replication after this. We copy them. Let me use, and you know what? Why don't I make this one red and this one yellow. So we copy them, but then we stick them together. Do you see the copy? So before you leave, I know it's time to leave, but look, before you leave, do you see the copy? So when I say copy, I mean they're copies. Bless you. The same genes, the same versions of the genes in the same place. These are called, before you go, please don't wrap stuff up. You're not taking notes. I don't understand how you're going to get this. These are called sister chromatids. Sister chromatids are identical. They're the same. So, and we'll talk about when that happens, but that's the key is that this is our objective, to copy the DNA in S phase and stick them together. This is during S phase. There it is. That's the idea, but that's not, so that's what happens with copying the DNA. Now, to get there, I'm going to zoom out again. I'm going to look at one cell. Here's a cell from a parent. To be clear, we call these gametes. I think it's... So gametes. Gametes are cells with 1/2 the DNA. So you start with 2 sets of chromosome, one from mom, one from dad. And we go through S phase and we copy them. We have sister chromatids. The first phase, and we're going to call them 1 and 2 because we're going to do it twice. We're going to half it by doing dividing the cells twice. That's how we're going to half them. The first division is just is separating the sets, and then the second division we actually separate the halves of DNA. So it's going to be 2 divisions. In the end, we're going to end up with 4 gametes. Our objective, gametes, but 4 of them. So what is the first phase of mitosis? It's exactly the same phase. The phases are exactly the same. What's the phase? What is the first phase of mitosis? Anybody remember? I'll give you 5 seconds and I'm going to answer. Prophase is correct. Prophase is the first step. And in prophase, you're going to condense the DNA. The nucleus is going to start to dissolve. So these two were in the nucleus. You're going to have the centrioles show up on both sides. They've already been doubled. The spindle fibers are start to form, right? So you start to get rid of the nucleus, starts to go away. Your DNA has already been doubled. The DNA is already condensed, wrapped around histones, and stuck together. So after prophase, what occurs? Metaphase is correct. So, and by the way, we call it prophase 1 in meiosis because it's going to do it twice. After prophase, we have metaphase. And in metaphase, what's happening is you have the spindle fibers already formed. They're already connected. The centrioles have connected them. Remember, you need the anchors. The centrioles are the anchors upon which the fibers act like cables. And you're gonna have you can actually see it in the microscope. If we have time, we'll look at them on Monday on Thursday in the microscope. The spindle fibers divide, give you a way to place the chromatids, so assist the chromosomes so they can separate. Now we line them up. They line up. Metaphase, the chromosomes line up. Their kinetic core, the center that connects them, is going to connect to the spindle fibers. And the sister chromatids, let me not use white here. The sister chromatids are identical, are going to line up. Again, I'm only drawing one set. This is going to happen with 23. It's the center. This is the kinetic core. It's a centrosome. It's the thing that connects the two sister chromatids together, sticks them together. Remember, I said they'd have to be stuck together so they move together. You don't want to mix them up or lose one, so you stick them together, like a leash on a dog, right? Like I don't know what other examples you want to say. Does that make sense? Okay. Is everybody okay with that so far? This should not be new. This is all should be old hat.
It's me.
Oh Jesus, you're trying to scare me.
Sorry. Here, I'm just returning it to you.
Oh, I'm so sorry.
No, it's fine.
I kept meaning to get it. I've been down to your room several times, but
It's okay.
so again, remembering that here on these chromosomes, you have these identical placements. And this is the way they would normally line up in mitosis. So this is what would happen in mitosis. In metaphase of mitosis, this is what would happen. Could someone close the door? I don't know why. I'm sure it's something very funny, but in metaphase of meiosis, something else happens. Very interesting. Here, the objective is to get one, is the skittle example of having to divide them the same on both sides because you want the same for mitosis. So you want this sister chromatid, this exact copy, moving over here, this one to move over here, this one to move over here. So you're going to have one copy from mom and dad on each one. We're good. Right? You want to keep them the same. Here the objective is to mix up the DNA. to recombine the DNA. DNA recombination is the key. So this does not line up in this way. Here, I'm just going to go back for a second and look at the original so I don't confuse you. Big A, big B, and little a, big B. Somebody remind me of that. So here, the two sets come together. They stick together. Two sets. So 4 sister chromatids. You know what? I should draw a different color, shouldn't I? It's going to be a real mess. 2 sets. So 4 sister chromatids, all. What's a prefix for 4? A prefix for 4. Like tetra is, tri is 3. What's the? It could be quad, yeah, but it's not quad. Anybody have another? 4. A prefix for 4. Take the headphones off your head. There's 3 people with headphones on their heads. Take them off. Tetra is, I already said it by mistake, but it forms a tetrad. So tetrad. This is a tetrad. This meiosis. Meiosis, you'll see why that forms that. In mitosis, you want to keep them separate. You want to line them up. You want to separate the chromatids. And you want the same chromatids on each side. Since these are all identical, everybody's good. Here, and I really should have done this a different color as well. I'm just trying to keep it neat and clear to you and what's going on. So let me go ahead and color this red as well. So in the end here, in mitosis, you'll have 1 blue and 1 red chromosome in each new cell. Do you agree?
Yeah.
This one's going to move in this direction. That one's going to move in this direction on the cord. You have 1 blue, 1 red, 1 blue, 1 red. This one, we want the DNA to be mixed up. That's like a major point. So in meiosis 1, metaphase, during metaphase 1, and it's really not in metaphase, it's actually in, unfortunately I didn't do it in prophase. It's actually in prophase 1 that this happens, so let me draw it up here. All right, so when the tetrad is first formed, when you first form the tetrad, actually this is very serendipitous. I used it again. When that tetrad is first formed, those 2 sister chromatids stuck together by this centriole. A centrosome, sorry, I keep using those 2 words are too similar for me. So this tetrad is formed during prophase 1. So they condense, they stick together, chromosome 1 from mom, chromosome 1 from dad. They have to be the same chromosome. I hope you understand that. And this is another reason why it's important that their genes are in their right place, in the same place. But they're going to stick together. Now, that by itself isn't a big deal, except the next thing that happens, can anybody guess or remember? So the next thing that happens is crossing over. So when they're stuck together, they're randomly going to get a piece being deleted from here and a piece not deleted, but they're going to switch. They're going to stick together. They're going to switch, and you're going to get a piece of that chromosome sticking over here and a piece of this chromosome sticking over here. So now you got 4 different chromosomes. Does that make sense? 4 different chromosomes. So this, again, this would be a piece of here, and this would be a piece there. Where you used to have 2 identical chromosomes, or sister chromatids, now you have 4 different sister chromatids. And in metaphase 1, they line up as a tetrad, and they're lining up this way. So in order to separate them, what you end up with is 2 cells at the end. Well, there's metaphase. What happens after a metaphase is anaphase, correct? And again, pretty much those were the big differences between meiosis and mitosis, because now here's another big difference between meiosis and mitosis. In mitosis, you separated the sister chromatids in anaphase. In meiosis, you're separating the sister chromatids. In mitosis, you're separating the pair. You're separating the pair. So here, as the kinetochore drags on, it's going to pull this pair. They're still stuck together. Do you see that? They're still moving together. Remembering that this one now has that going on, has a little bit of the other chromosome on it. And obviously the pair of sister chromatids, the other pair moving in the opposite direction. Just drawing the white line so you can see a little better. So one's going in one direction, the other's going in the other direction, remembering, of course, that this also has a blue tip. So then, after anaphase, you have telophase, correct? Correct? Yes, no? Telophase.
Is that anaphase with meiosis?
Anaphase 1. If it says 1, it's meiosis. And in anaphase, you see these are not single chromatids. They're still stuck together. They're still paired up, and they're moving. So you're still going to end up in telophase. In telophase 1, what you're going to end up with is a cell with, right before it divides, it's going to have 2 nuclei. Two nuclei. In one nucleus, you're going to have 1 chromosome from mom that's still mom's chromosome, and one sister chromatid from mom that's that's the original. 1 sister chromatid that has a little bit of dad's in there. So you have this mixed up DNA already, and we haven't even mixed it up even further. We're going to mix it up even further, but we've already recombined the DNA there. And then, of course, on the other side, you have a chromosome number 1. Again, I'm going to keep emphasizing this. This happens to all 23 chromosomes. And the other side has that same chromosome number 1. Except that there's that little bit of recombination that occurred from crossing over. Is that making sense to you?
Yeah. So
So in prophase 1, everything's the same except you have the tetrad forming. 4, tetra means 4. Tetrad forming. That tetrad then in metaphase 1 lines up together. The DNA is already being exchanged, crossing over has already happened in prophase 1. And then anaphase, the pairs separate. And at the end of telophase, you still have 46 chromosomes. But the DNA has been mixed up. So you still have 46 chromosomes, DNA is mixed up. Now you have cytokinesis, and you have 2 cells. And inside those cells, each has a nucleus. I'm not drawing the other organelles. I hope you understand that. And in one cell, you have this doubled up. It's already doubled up. No need to double. No, we're not doing S phase again. There's no S phase. Because if we did S phase, it would defeat the whole purpose of dividing it. And so you have these 2 cells that are not identical anymore, but they still have 46 copies of the chromosomes. That's no good. So what do? How do we fix it? Split them again. Do it again. But without S phase. No S. We're skipping S phase this time. So at this point, you're going to do prophase 1, dissolve the nucleus. DNA is already condensed. Spindle fibers are going to form. Centrioles have been copied. It's amazing to me how fast this happens. Spindle fibers form. This time, though, they're going to line up just like they did in... They're going to do just what they did in regular mitosis. They're going to line up... I have to make this orange here. They're going to line up as sister chromatids. So this time, the sister chromatids are going to separate. Again, I'm only drawing 1, but this is going to happen 23 times. I put too many centrioles, but that's fine. And that happens for both of these OG original. The sister chromatids line up. The chromosomes line up with the sister chromatids going to separate. And so what you'll end up with that. And of course, this is prophase 2. There's no difference between prophase 2 and mitosis prophase. And then after prophase, you have metaphase 2. In metaphase 2, there's literally no difference between that and regular mitosis metaphase, except that you'll have half the DNA. And then notice that in metaphase of mitosis, when we look at mitosis metaphase, you have 2 chromosome number 21s. 2 chromosome number 1s. There's 2 of them. So when you separate the sister chromatids, you're going to have 2 chromosomes, correct? All right, so the difference is in meiosis, when you separate these sister chromatids, and let's just go ahead and do it. Let's go ahead and let's do anaphase 1, anaphase 2, which is the same as as regular mitosis. Again, I'm only drawing the nucleus. then what's going to happen is you're going to get the sister chromatids moving. One moving in this direction that looks just like the original, and one moving in the opposite direction. I wish I would have chosen a different color for this centrosome. One moving in the opposite direction. That's just slightly different because they're crossing over. And then the same thing happens in anaphase over here in this septic course. Except, of course, that in the end, in telophase 2, when we look in the nucleus, as the nucleus reforms, you're going to get one sister chromosome, one chromosome for each, so 23 chromosomes. You have 23 chromosomes. So this is going to divide into 2, by the way. And this is going to divide into 2. Does that make sense? Am I clear on that?
Yep.
Each of these, so that's 2. So you're going to end up with 4. One is going to have the original DNA. From mom. The other one's going to have the original DNA from dad. And the other is going to have, of each, is going to be mixed up. So now where you used to have 2 identical cells, now you have 4 different cells. Multiply that times 23. So a lot of genetic variation. Sex is all about recombining the DNA. In biology, that's what sex means. Sex is recombining DNA, mixing up the DNA. Not only are you mixing your DNA up with your partner, but before you mix your DNA up with your partner, this is all before, you've already mixed up your DNA. So you're maximizing the genetic, we call it genetic recombination, or the genetic variation. And we do that using recombination. Genetic variation is maximized by recombining the DNA. What do we call recombining DNA? In nature, we call it sex. That's what sex is. To a biologist, sex is recombining the DNA. All right, so that should help you, right, with what meiosis is. I walked you through the entire process, and here you'll see it all drawn out. And there's a really neat little pogio, and even goes into fertilization. It talks about a polar body being made, and we'll talk about that on tomorrow after your test. I'm thinking maybe you should just wait till the test on next week so we can finish meiosis. How do you feel?
Is that one of the
Yeah.
I don't have to put on the test. I can choose.
Oh, is that one the same?
Oh yeah, it's up here. So it's up to you. I can add meiosis to the test and do it next week and just put it all in one test, or how do you want to do it?
Put it on.
So on one test. All right, so we'll move the test to Thursday next week, and we'll just go over this, finish meiosis tomorrow. Do this tonight for homework. It's up here. It should be pretty easy. Yeah. It's a really good Pojo, by the way. Did I time it perfectly?